Hi everyone, in this video we're going to go over DNA replication. At the top here we can see our central dogma, which is DNA to RNA to protein. So, starting with DNA replication, we typically have our DNA which is coiled up, as you can see here, in its normal configuration. Usually we wrap around histones. But if we want to replicate our DNA, the first thing that we have to do is get it open. So we'll use Helicase and topoisomerase to do that. Helicase unwinds the DNA from this configuration. Let's just say we turn it sideways and open it up to this bubble kind of configuration. This is going to be called our replication bubble. Now, what topoisomerase does is prevents the supercoiling of the DNA. So I'm not sure if some of you guys know, but corded phones, like, they have a coil to them, and then they would used to coil on themselves. So in order to prevent that, we have to snip it at certain areas in order to release that tension, because built up tension degrades the DNA. As I said before, we have our helicase, which takes our DNA from this to this, and then we have our topoisomerase, which prevents the supercoiling of the DNA by snipping it in certain areas. Next up, at number 3, we are going to have our single-stranded binding proteins. I'm just going to abbreviate that by using SSBP. And what these proteins do is they bind to the free nucleotides of the DNA. And what they do is they prevent the nucleotides from reattaching to one another. So basically, it's like you have two magnets that are attracted to each other, and you stick, like, let's say, a big piece of cloth or even a chalkboard eraser in between them. Their strength and affinity for one another decreases so long as that object is in between them. And that's exactly how the single-stranded binding proteins work. Next up, we have our primase. And what this does is it lays down a first strand of not DNA, but RNA for us to work with. Okay. Now the top part is what we're going to consider to eventually be our leading strand. And the bottom here, which I'll get to in a second, is going to be referred to as our lagging strand. And I'll get to what those two things mean. But just for now, just know that primase is laying down the initial codons. Just remember that this is made out of RNA. Next, we'll have our DNA polymerase 3 come in. And what DNA polymerase 3 does is it adds the nucleotides in the 5 to 3' direction, as you can see down here. Made a little bit of note of that. We only make DNA in the 5 to 3' direction. We never make it 3 to 5. Now, I will say for Dr. Bazan's test is that you want to be careful about this, because we read the DNA, as you can see here, from 3 to 5'. But the new daughter strand, we make that in the 5' to 3' direction. Okay. So just remember, new DNA is made 5 to 3, while the old strand is read 3 to 5. Just something that you want to keep a lookout for when Dr. Bazan gives his test. It can get a little bit wordy. So just remember that. So it adds the big chunk of the nucleotides at the leading strand and here at the lagging strand. Now let's take a closer look at the lagging strand. So here we have our lagging strand with our primers put in already by primase. Now the reason why we have to do this is because DNA is antiparallel, and what that means is that we have two identical strands that are running in opposite directions of one another. So what that means is we have our 3' here and our 5' here. That means this new daughter strand, the 3' end and the 5' end, go to the opposite ends of each other. So the 3' goes with the 5' and the 5' of this strand goes with the 3' of this strand. As I said before, we can only make DNA in the 5 to 3' direction, which is why we can't go from here to here, because this new strand then is going to be running from 3 to 5. So the way that we do that is we give ourselves several primers, and then we're able to lay down new nucleotides in the 5 to 3 orientation. And that's going to be the same for all of these. So 5 to 3, 5 to 3. Now that we have all our nucleotides in place, we're going to next use DNA Polymerase 1. And what DNA Polymerase 1 does will remove the original RNA primers and insert regular DNA nucleotides. Remember, the thing that separates RNA from DNA is an OH group. How come we can't have RNA in our DNA? Okay, something that you'll want to look up, and it has to do with what organisms use RNA as their template strand. Okay, so we use DNA. The question is, what uses RNA? If we were to use RNA in our DNA, we would see it as foreign and attack it. So if you guys want, feel free to answer that question and shoot me an email. Moving onward, let's take a closer look at how DNA Polymerase 1 works. These red here are indicating DNA nucleotides, while the green is still the RNA nucleotides. We would have our original primases here, but RNA polymerase, what it does is it'll kick out the RNA and add in its own nucleotides, its own DNA nucleotides. However, as you can see here, there's a small gap in between the DNA polymerase laid down by pol 1 and the DNA nucleotides laid down by DNA polymerase 3. So now that we have our RNA primases removed and DNA nucleotides put in, the last thing that we have to do is to join the phosphate backbones, and we do that by using ligase. Now, ligase doesn't add any nucleotides. All of that was done by DNA polymerase 3 and 1. What ligase does is it just simply joins the phosphate backbone. So let's take a quick look back at how DNA pol 1 added nucleotides. Remember the gap that we talked about here? That will then be taken up by ligase. Ligase just joins that phosphate backbone. And if you want a more realistic look to it, how this would look is nucleotides here that have their backbone. And we also have the nucleotides from DNA polymerase 1 that have the backbone, but they're not joined together. DNA polymerase 1 can't do that, but ligase can definitely do that. Just as a brief overview, I hope you guys can see that the yellow here is in between the red and the blue. That's just to show that we've joined that backbone. We have our DNA originally wound up. It's taken apart by helicase and topoisomerase to give us this configuration. Again, topoisomerase prevents the supercoiling of the DNA further down from the bubble. We use our single-stranded binding proteins here in order to prevent the hydrogen bonds reattaching the nucleotides. RNA primase comes in where these red boxes are. They give us an original template to start with. So DNA pol 3 cannot start adding nucleotides on its own. It needs somewhere to start. It's kind of like that spark to a fire. So primase will lay down the primers from which DNA polymerase 3 will start adding nucleotides. After that, we have our DNA pol 1 that will then pop off the primases here and replace them with DNA nucleotides. However, DNA polymerase 1 cannot add the phosphate backbone. That's what we have ligase for in order to join the nucleotides that were laid down by DNA polymerase 3 and DNA polymerase 1 in the lagging strand. And as always, the leading strand is made continuously, while we have our lagging strand, which is made in segments called Okazaki fragments. And DNA is always made in that 5' to 3' direction. This is why it's made in segments, because we lay down the primers so that the DNA polymerase 3 can travel in that 5' to 3' direction, because it can't go this way. Alright, if you guys have any questions about this, as always, you guys are more than welcome to shoot me an email and ask questions during office hours.